Good morning, students. Today we are going to study the chapter Sexual Reproduction in Flowering Plants. You're already thorough about what reproduction is in your previous chapter, Reproduction in Organisms, and you are already understood about the features of sexual reproduction, including the events of sexual reproduction. So, once you're clear about it, now we can study the sexual reproduction in flowering plants. So, first you should know what flower is, how it looks, what it forms as on a plant. So, you find it in angiosperms, and it is a fascinating organ they've given. Of course, it attracts, it has a beauty in it. Okay, so it attracts to you. So, what is that fascinating thing? It is used for various things like for worship. Maybe you can take it as your religious, aesthetic. Then there is ornamental for decorations. It may be like you're decorating a house with different flowering gardens, okay, flowering plants, or it may be like vases they keep. And you might have seen when even in cultural, you can say dance program and all, you all use flowers on the head, or any like celebration comes, you all wear flowers. And then social, I'm trying to say about the national flower here, the lotus. So not only the lotus flower, even the daily use. Some people use, they know they have that tradition. The girl has to wear a flower on their head. So you can say the society will indicate the girl will only wear flowers. Boys will only offer flowers to girls. So it may be that way. Okay. The various, there are many ways you can explain about how the flowers can be used. So I hope it is understood. It may be of religious worship, cultural value, ornamental for decorations, okay, and social. What does it symbolize? It symbolizes to convey human feelings. What are those feelings? You can find affection, grief, happiness, mourn. So affection to show love, grief to show sadness, okay, and again happiness, mourn. You might have seen even in during funerals, it is one way to offer flowers to say goodbye. Okay, so these are some photos I've kept for you to make you get the picture how the horticulture. This is the place where the flowers are grown, made them become very healthy. You find hybrids. So this is a place, we call it as your horticulture. So I hope you can get how the feeling comes here when you offer a flower, when you decorate, okay, when you this is one way of decorating. It is part of your preparing a vase. Then there is your the garden pots, flowering pots. Then you have your field, the sunflower plants, the ones which we are very much attracted to about the sunflower. Then marigold. Then you have your lotus here, the national flower. So you all can notice here what I've kept here. If you all notice it, that this portion, it is your flower stall. The garlands are there. So you all know very well where they are applied. I will not go in detail. Just talking about the flowers, where and all it can be used. So it's coming under floriculture. Why floriculture? Horticulture includes everything. It can be your vegetable plants, it can be your croton plants, you can get your flowering plants, But we are talking about especially about the flowers. We need flowers. There are many. You might have seen certain shops where they prepare bouquets and all. So those flower shops, they do one kind of business. Only flowers they sell. They don't sell the plant. So from where do they get the flowers? It is only through floriculture. Okay, floriculture, another term is flower farming. So this is not an important definition. It is not important. So this is an all you can just strike off. Only to know what it is. It is just a flower farming. So it is concerned with the cultivation of flowering and ornamental plants. This is important. Where and all it is used. and which type of flowers are used. So ornamental, there is your rose, there is zinnia, gerbera and jasmine. You all know very well during a function, gerbera is the one which you have already seen before, giving you idea. See, observe here, this is in the garden. Okay, at home, you all grow it in your garden, which we say zinnia. Okay, and this is the one. Let me change the color for you. This one is the gerbera, which you most commonly find in your most of the functions as a decoration. Okay, this is your chrysanthemum, sevantige we say, your jasmine and the lotus flower. I've kept few for you to get the idea. So social use, there is lotus, there is jasmine. Then cultural use again, jasmine, chrysanthemum, marigold. So you have got the idea now. So now we can say what flower is. It is the morphological and embryological marvel. Meaning of marvel I've given here, a wonderful creation. Okay, and it is the site of reproduction. Very important is it is the site of reproduction. Dealing with the structure of flower, this is the LS of flower, can be asked for 2 marks. Draw a neat label diagram of LS of flower. LS is longitudinal section. Okay, so you all know about the 4 whorls of the flower. There is your sepal, there is whorl is your calyx, petal is individual unit, whorl it is your corolla, then there is stamen and pistil. So these are your 4 whorls. Among these you can find here, I've written in red the stamens and pistils. These are the units of reproduction. Okay, after photoperiodism, flower will develop from the flower primordium. Now this sentence is trying to say a flower can develop only after the induction of a light period. You all remember photoperiodism? A duration of your daylight and also a duration of night period. So this together will induce the flowering process. So usually it is here your normal, the plant, okay, that is only having the leaf primordia. So it gives rise to your shoot. Whereas your photoperiodism will give rise to your reproductive phase. What is that reproductive phase? The flowering period. Okay. The flowering period, you all can observe here how the flower meristems in blue, which they are showing, you can observe here in blue they are showing, is the one which is going to develop into the flower. So that is your flower primordia. The blue color structure we call it as flower primordium, and that will lead to the formation of the flower, and it is possible only after a period of light. That period of light provided is called as photoperiodism. This you have come across in your first PUC. Okay, this it's only explaining, this diagram is not important. Okay, it is only explaining this sentence over here. Okay, now recalling back your 3 events: pre-fertilization, fertilization, post-fertilization. Here we are going to study one by one, and under that we are studying the structure as well as the event. Okay, so in plants we are studying the structure of the flowering part, the flower containing that sexually, I can say it as a part for sexual reproduction. That structure we are studying in detail. and the event occurring within that part or within that sex organ. So this is in flowering plants we are studying. Similarly, in your post-fertilization, there is a structure and event. Whereas fertilization, we are not talking about any structure, it is only the event. Without any delay, I am starting with the first one, the pre-fertilization event. Please note what and all we are going to study in pre-fertilization event. Okay, so there is your structure, that is stamen, then there is your microsporangium, then there is pollen grain. Okay, so this is the male sex organ, completely male sex organ. Okay, and this is the structure, the event is microsporogenesis. Okay, I repeat, under male, okay, under male sex organ or the male reproductive part, we are going to study the stamens, the microsporangium structure and the structure of pollen grain. Okay, this is only the structure part, and in that, especially in the sporangium, the event is microsporogenesis. Okay, you should remember this. So this is for male reproductive part. We are going to study in detail the male reproductive part first. Just to know in advance about the female reproductive part, it consists of pistil, it consists of megasporangium and embryo sac. Okay, and in your megasporangium, megasporogenesis occur. So this is your structure, this is the event. Okay, so we are going to study the stamens, microsporangium, pollen grain structure and microsporogenesis event now. Starting with your structure and events in stamens. So just observe the stamen. You all already studied before. Stamen consists of anther and filament. So it consists of the two parts. Remember, stamens have another name. The technical term given is andrecium. Remember it. And the anther is called as your bilobed structure. And filament is called as your long slender stalk. So based on the structure, you can even compare over here with the diagram. Whatever the dots which are shown, it is your pollen grains. Don't worry now. If it is asked for 2 marks, you should be able to draw the stamen. The whole structure is stamen. The shape and the size of stamens will vary in different plants. So you can observe here. Look at the shape and the size. It is varying in different plants. So based on the attachment of stamens, this is a spelling error, it is stamens. The attachment of filament actually, the filament is only this part. You can just observe the blue part. Okay, observe that. Now okay, now coming to your portion. Based on the terms, they've given the first one adnate, meaning to say it is running along the two bilobed structure, the anthers. Okay, along the anther. Basifixed, it is stopped here. So at the base of the anther, it is connected. Adnate along or across the anther. Okay, basifixed, at the base of the anther there is the connection. Dorsifixed, it is in the middle portion. The middle portion is your dorsifixed. The filament is attached to the middle portion. And versatile, you can see it is exactly in the middle. Okay, here let me tell you, there is little attachment downward also, little attachment downward, so that it is firm, the position is firm. Here it is only in the middle. not even going a little aside for the connection or for the support. So this can easily move up and down like a seesaw. So that is why it is called as versatile. So I'll repeat again. The filament attached throughout the anther from the tip till the base, okay, that is called as adnate. Basifixed from the base of the anther, the filament gets attached. Dorsifixed from the middle, it goes little down attached to the anther and then form as your filament. And versatile, only attached to the middle point, making the anther free for movement. Whereas these three are not free for movement. Only versatile, it is free for movement. So this can be asked for 2-mark question, and if it is asked for 3 marks, see that you are going to draw. Okay, so it is based on the attachment of filament to anther. Please remember, make a correction here. Attachment of filament to anther. So based on that, the stamens are classified into 4 types. Based on the number of stamens now, based on the number of stamens, we are studying, there are different types here: monoadelphous, diadelphous, polyadelphous, synandrous, syngenaceous, polyandrous. To understand one by one, first let me tell you, adelphous means bundle formation. Okay, remember that, bundle formation. Look at this. You can find the hibiscus portion here, and that has all over here the pollen grains, sorry, not pollen, anthers, the yellow color portion, and these are your red color portion. So the yellow color portion, all those are anthers, and they are all fused to form one filament. All are fused, filaments are fused, anthers are free. Even here you can find there are 2 bundles. This is one bundle. Many, it is countless, you cannot count it. In hibiscus there are many anthers which the filaments fuse to form one bundle. That's why the term monoadelphous. Here there are 2 bundles. The filaments form to produce 2 bundles. Okay, again here anthers are free. So here this is your diadelphous condition. And polyadelphous, you can see there are many bundles. Many bundles. See the anthers. Anthers are free here. So filaments are fused to form bundles. More than one bundles are called polyadelphous. So adelphous means bundles. Here it is one bundle. Here there is 2 bundle. There are 2 bundles. And then here there are 4 bundles. So those 3 names you have got: monoadelphous, diadelphous and polyadelphous. Now syngenaceous, you can find here the anthers are fused. Here it was all filaments that were fused. Now anther is fused over here. Anther is fused, filament is free. Observe here, filament is free. Synandrous, here both anther as well as filament is fused. Observe carefully. Anthers, there are many anthers, and each anther has a filament. So all the filaments fuse to form one bundle, and all the anthers are forming one bundle. So together everything is clubbed, and the term is called synandrous. Syn means fused. Synandrous. Polyandrous, your free stamens are called as polyandrous. They are free. They are not fused in any style. They are free. Okay. So this is your polyandrous. So this is based on the number of stamens present in the flower. In one flower is having so many types here. Based on bundle formation, 3 types. And again here, syngenaceous, we are talking about the anthers which are fusing. Synandrous, both are fused. And your polyandrous, there is no fusion at all. It is totally free. Adelphous, only filaments So monoadelphous, diadelphous, polyadelphous. These are three types where filaments are only fused. Anthers are free. Okay, without any delay, I'm going forward. Let us understand about the anther. First, look at how anther, the structure is. Look at the structure. Okay, look at the transverse section. If you all wonder why two structures are there, this is the young one. This is young. Okay, and this is mature. The down is mature. Observe carefully. I have taken the section and the section is shown here. We are studying this part in detail, not this. The young anther only. Clearly observe, there are 1, 2 in one side, 1, 2 in other side. So lobed structures you have to observe. 2 are here, 2 are here. Totally it is 4. Okay. But we are talking about one section each. And you can observe this part, the round pink portion. That is again an important structure. And we'll be understanding one by one. Oh, again, even this one you have to observe. You can find a slit here. You can find one slit. The split over here, which releases the pollen grains. That slit is actually here. It's actually here, at this portion. Why I said at this portion? You can observe here, the split has opened here. When it is mature, it opens here to release the pollen grains. So I hope it is clear about the structure. Now going in details about the parts. It has 2 lobes, hence the term bilobed. Anther is bilobed. Okay. Each lobe contains 2 theca. So coming to that term, 2 theca, and hence the term dithecous. Remember, each lobe. So we are talking about one lobe over here, another lobe over here. Okay. You can find here circles. This is your microsporangium. That microsporangium we call it as theca. Okay, theca we say, because that theca has one more layer around it. That's why together along with the wall layer it is called as theca. Only the pink portion is microsporangium. Okay, so don't get confused. Here there is 1, 2 structure. It looks like a lobe. Here also there is 1, 2 structure. But here we are dividing, as this is one lobe, this is another lobe. Okay. Even when you draw the anthers, we draw it in the same way. Let me just show you the picture here. See, observe here. We show it as 2 lobes. And inside each lobe, there are 2 thecas. That's why this theca is this portion. The pink with one layer of a layer of cells. So I hope the word is clear. Anther is bilobed and each lobe has 2 theca. Each lobe has 2 theca. That's why the term dithecous. It has a longitudinal slit or longitudinal groove separating the 2 theca. Now you understood, it is a slit or a groove that separates the 2 theca. So whatever I've told you, it looks like a lobe here. In between there is a depression which later forms as your longitudinal slit. Why it is a slit? It opens up during maturation to release the pollen grains. Okay. So when it is young, it is together. When it is mature, it will split up to open to release the pollen grains. So the terms are clear: bilobed, dithecous, and the longitudinal slit or longitudinal groove. Coming to the TS of young anther, now remember, This is the TS of young anther. We are going in detail. Just before we go in details in the theory part, let me make you clear with the diagram here. You can see here something, it is dark. So that little dark portion is your connectivity, where vascular bundles are present to send nourishment to the anthers, okay, to send water to the anther. So that's why there is a vascular strand which we call it as connective. Then this particular pink portion is microsporangium. Each is microsporangium. If you count all four, it is microsporangia. Okay, plural. So there are 4 microsporangia. Okay, surrounding it is one layer. You can see. Can you see one layer of cells? They are uniformly sized. Those are one layer which is nutritive and nourishes the microsporangium. And followed by that is your certain other, you can see 3 to 4 layers of thin or small sized cells. Then a pink layer again you can find, which we call it as endothecium, and the outermost again a very thin and small sized cell, it is the epidermis. Okay, so you can see here they have shown endothecium, they have labeled it, epidermis is the outermost. Middle layers, I've told you just now, this 3 or 4 layers which is small. And last the tapetum, which I said, which is surrounding, I've told you this, nourishment, the surrounding cells surrounding the microsporangium, those cells are nutritive, okay, and they are called as tapetum. And this microsporangium, which I said it is pink in color, consists of sporogenous cells. So you have got some idea there. Without any delay, I'm going with the features now. If it is asked for 5-mark question, you should be able to answer the question here. So this is the structure part. The TS of anther is tetragonal or 4-sided. I hope the word is clear. Tetragonal or 4-sided. Remember I told you 1, 2, 3, 4. Let me go back to make you clear. I told 1, 2, 3, 4. Why did I say that? It is for the structure. You have the shape as tetragonal. What shape? When you take the section, it is 1, 2, 3, 4. It is tetragonal shaped. If you see as a whole, it is bilobed anther. If you take this section, it is like a butterfly there. Okay, so it has your 1, 2, 3, 4-sided. We say tetragonal shaped. Okay, what does that mean? Tetragonal. It means it contains 4 microsporangia. Okay, at the corners. That means in each lobe how many? Yes, 2 microsporangia. So just to recall back, there are 4 over here, 4 microsporangia. I've divided to show one lobe, another lobe. So in each lobe, how many are there? 1, 2. Okay, so we are saying 2 microsporangia in each lobe. This microsporangia will develop into pollen sacs. This we are talking about development is when it matures. Okay, I'll show you back the figure again. When it is young, it is your microsporangium. When it is mature, you can observe here, it is called as pollen sac. Why pollen sac? Pollen grains are already inside it. Okay, so before it is called as the theca. Okay, that means microsporangium with certain layers of cells around it. That is your theca. This microsporangium later, only the pink portion, develops into pollen grains. So that particular theca now turns into pollen sac. Remember, the theca, meaning to say the layer of wall cells are there. Wall layer cells we say. They are still present. You can observe here. Okay, and the endothecium is thin over here, that's why it splits up to release the pollen grains outside. So it's clear here now. So microsporangium, the pink color region, has developed or matured into pollen sacs, and pollen sacs are packed with pollen grains. Again go back and just observe. This microsporangium develops into pollen sac, and pollen sacs are already packed with the pollen grains. So it's already pointed here, pollen grains. The terminologies are clear here with the structure, I hope. Again, this is your very important diagram. This particular diagram is important for you. It is for 3 marks separately asked. Draw a neat label diagram of TS of microsporangium or TS of young anther. This is young anther. Whatever down it is, there is the mature anther. So you observe this, it is like an outline here, and one zoomed portion is shown here in clear. I want everyone to do it very neatly. Don't take it very lightly. Do it neatly because it is a 3 mark question and this chapter has a lot of weightage. So please do focus on diagrams also. Remember, whatever you can find here, the labelings, make sure you are writing in your capital letters. Let it be in pen, let it be in pencil. It doesn't matter. Diagram, you keep it in pencil. Okay, labeling maybe pen or pencil. This is for exam. For lab records, it is only pure pencil. You all know already. Okay, so see that you are making a practice, because sometimes in pen error comes and you have to strike and rewrite again. So for that reason, we recommend you can use only pencil for labeling. Okay, so you remember, this is the diagram which you have to follow from your textbook. Do it neatly and submit it. Coming in detail about the microsporangium structure now. Okay, so remember, this is the TS of young anther. TS of young anther. We are now going in detail with microsporangium. Now the question comes, now where is microsporangium? Again going to recall whatever the circles you can find here, those are your microsporangium. So you can see here, those are your microsporangium. This is the portion. So giving you a better idea what we have started with, you can see the pink portion. Pink portion only is the microsporangium. Okay, there are 4 microsporangia. Remember the terms. Without further ado, I am going in detail with the structure of microsporangium. Appears circular in outline. Okay, just recalling with the structure, I am referring to this diagram now. Look at the outline. This is only the outline diagram. Can you observe the circles? That circular outline is your microsporangium. So appears circular in outline. It is surrounded by 4 layers. So remember this, I had pointed before the 4 layers. The outermost is epidermis, followed by one single layer endothecium, followed by middle layers, 3 to 4 layers of middle layers. Okay, and finally the tapetum. The first 3 layers, that is epidermis, endothecium and middle layers, they have a function, and that is to perform protection and dehiscence action. Okay, protection to keep the microsporangium safe, so the development of pollen grains occur. Dehiscence, it is the release of pollen grains when it is matured. Tapetum, very important, for 1 mark it can be asked. Give the significance of tapetum. It is a nutritive tissue. Remember, tapetum is a nutritive tissue, and this tissue contains dense cytoplasm and more than one nucleus. What is the main function, main significance? It nourishes the developing pollen grains. It nourishes the developing pollen grains, the ones which are forming into pollen grains. Okay, so there is your 4 layers can be asked separately as your 2-mark question. Now studying the event. Look at the word here, microsporogenesis. So remember the term microspore and genesis. So microspore formation. Okay, microspore formation. So look at the term microsporangium. Look at the term microspore. Can you recall back to your first PUC classes? The sex organ was called as sporangium, and whatever gamete we got was spore. Okay, or haploid structure is called as a spore. Okay, so similarly we are talking about in case of your microsporogenesis, we are saying microsporangium which consists of sporogenous tissue. So what is that sporogenous tissue? They are the group of compactly arranged homogeneous cells. This you have to memorize. 1-mark question. What is sporogenous tissue, or what does microsporangium consist of? So the answer is sporogenous tissue. It is a group of compactly arranged homogeneous cells. Compactly, they are like close to each other, not leaving any space. Homogeneous, all are alike. You are not able to find out as this is one type of cell, that is another type of cell. They all are alike. Such cells are called as homogeneous cells. And this sporogenous tissue is the one which will undergo meiosis to produce microspore. Just look at the term microspore tetrad here. It's given as microspore tetrad. The term only says tetrad means number 4. So 4 microspores are produced. I said meiosis. Just coming to this particular picture first. Observe this microspore mother cell, or you can say the sporogenous tissue having the homogeneous cells. So that particular cell undergoes meiosis 1, meiosis 2. So you can find it first forms a dyad and then tetrad, meaning to say there are 4 haploid cells. So coming to the definition, it is the process of formation of microspores from pollen mother cell through meiosis. So what is that definition trying to say? There is already the pollen mother cell. You can find here all these are sporogenous cells. Sporogenous tissue is the group of cells. Okay, and each cell is called as your microspore mother cell, or you can say pollen mother cell. There are 2 terms given, microspore mother cell or pollen mother cell. So you have understood about the microsporangium consists of sporogenous tissue. So another name for sporogenous tissue I have shown here, microspore mother cell. I hope it is clear, which gives rise to microspore tetrad. Okay, that is what is clear over here. Microspore mother cell, and through meiosis it is reductional division. So here you finally find haploid. This was diploid. Microspore mother cell was diploid. On meiosis it has produced haploid, 4 haploid cells. 1 diploid mother cell has given rise to 4 haploid microspores. So this definition is clear. Formation of microspores from pollen mother cell through meiosis. Very important is that meiosis term. Okay. So the cluster, what is 1-mark question? What is microspore tetrads? Okay. The microspores formed through microsporogenesis are arranged in the cluster of 4 cells. They are arranged in the cluster of 4 cells. Hence the name microspore tetrad. That's the answer. You have the question at the end. Please answer similarly as I said. Okay. when it matures, so you can find the term anthers when mature and dehydrated, the tetrad will separate to form pollen grain. So microspore tetrad will separate. So what is that microspore tetrad? Observe this. This is the microspore tetrad. Each will separate. Okay. So I am showing this direction. One will come out separately. It will modify itself and develop into pollen grain. This particular changes I will explain in the pollen grain structure. Okay. So please remember, when the anther dries, so as the anther matures, you can find the dehydration process leading to the separation of the 4 microspores, and each microspore develop into pollen grain. So from each microspore we are getting a pollen grain, the male gametophyte. So going back to this sentence here, the tetrad will separate, you have understood, forming pollen grains. On dehiscence, it releases several thousands of pollen grains. So this is, I am going back to your first picture. Let me show this picture here. This is your mature anther. You can find how the pollen grains are coming out during dehiscence. So this is your dehiscence process. Observe here, it has opened up, the longitudinal slit has opened up and releases the pollen grains to the exterior. So many pollen grains are there. Okay, several thousands they have mentioned. So it's clear now. Let me show you another picture here. This is from your textbook. How the microspore tetrad, there are 4 haploid. Do not write 4n. Remember, it's always 4 haploid. We show that bracket that way. Do not write 4n. 4n means tetraploid. We don't want tetraploid condition here. We want only diploid and haploid words. Your microspore is all haploid. 4 haploid microspores. Each haploid microspore develop into pollen grain. Okay. So it's clear. This particular explanation will be again coming under pollen grain structure. So just summarizing this microsporogenesis process, because this is a 5-mark question. This is a 5-mark question. Please remember, it is important. So I am summarizing it. It is the process of formation of microspore from pollen mother cell through meiosis. It's called microsporogenesis. Here the sporogenous cells divide meiotically, okay, to form microspore tetrads. So in this process, haploid microspores are formed. How many? 4 are formed. That's why it is microspore tetrad, a cluster of 4. Where does it occur? It occurs inside the microsporangia, or another term, pollen sac. This you better keep it for mature state, microsporangium for the young state, just to avoid any confusion. Okay, so there are 4 microspores formed, arranged in tetrad, and all are functional. All 4 microspores are functional. This is important. Okay, so all will develop, all 4 microspores will develop into functional pollen grains, and each of these pollen grain is termed as male gametophyte. That's why the last sentence says microspore gives rise to male gametophyte, which is nothing but your pollen grain. Okay, so this is in one picture summary. You can see how young anther is and the mature anther is. This is how your one lobe, you can see the division, one lobe having two theca. Bilobed condition, I hope it is understood, 1 and 2 lobes. What is dithecous condition? You can see the 2 thecas. What is that yellow portion? You can see over here. It is the microsporangium. Okay. If you all see this microsporangium structure, it is circular in outline. Observe, circular in outline. What are the 4 layers of surrounding the microsporangium? The 4 layers surrounding the microsporangium. There is epidermis, you can find it here, epidermis, endothecium, middle layer, tapetum. So please remember the sequence. Outermost is epidermis, next is single layer endothecium, 3 to 4 layers of middle layers, and nutritive tissue tapetum. And whatever the yellow portion is there, this one is your sporogenous tissue, which develop into your microspore tetrad or male gametophyte. So you can find how middle layer and tapetum disintegrate later on, and it will help in dehiscence. So it is clear in one picture itself. Please answer these questions and send it to me. Yes, thank you students.